Welcome back to Apoptosis series. This is episode 5. I am Dr. Kalpana Rakholia and today we are going to discuss about biochemical hallmark of apoptosis process. So before we discuss various kind of pathway, external or internal pathway, we must to know about which kind of biochemical hallmarks or events will be going to occur in apoptosis process. So these are the various points which we are going to discuss in this presentation. First of all, Procaspase is going to activate into caspase, which lead to activate the various kind of other events like it activate the CAD. Then DNA fragmentation will be occur, chromatic going to condenses, then membrane blabbing will be occur, and apoptic body formation will be done after formation of apoptic body. The cell membrane of this apoptic body will occur various kind of changes will occur like a phosphatidylserine externalization will occur, so it will give the signal. It means that's why it's the next activate the macrophage that will going to phagocyte the small apoptic body, and afterward apoptosis process will be occur. So let's discuss about how activation of caspase will occur. So caspase are cysteine aspartate. Full form is Cas. It means cysteine aspartate protease synthesized as an inactive procaspase. Inactive procaspase is known as zymogen. They have a no functional. When it are going to activate, then it called as a activated caspase. So inactive caspase also known as procaspase or inactive caspase. Then it are going to activate it, and it's known as activate caspase. So activated caspase or they are activate initiator caspase. There are two kind of caspase: initiator caspase like 8, 9, 10, and executor caspase. This initiator caspase activate executor caspase like 3, 6, 7, and ultimately the various kind of events will be occur. How it will caspase are going to activate? So in detail we talk about that. Inactivated procaspase which contain amino group, one side is a carboxylic group, and there is a large subunit and small subunit, and upper layer upper part is a prodomain, right? So these are the inactive procaspase. When activation signal are going to activate during the apoptosis process, either it may be signal may be intrinsic or extrinsic. That how this signal will be occur, that we already discussed in previous episode. After the signaling, that this caspase are going to dimerize. This caspase are dimerized and come together and cleave. This dimerization caspase going to close together, and here a specific cleavage will be occur at this site. So aspartic at the aspartic residue, and then after this prodomain part is removed, and these are the going to activate. These are the tetramer caspase that are going to activate, and this activated caspase going to target various part, and according to that apoptosis process will be occur. So in the summary, we can call that there are caspase are cysteine aspartate protease which are synthesized as an inactive procaspase. We also called as a zymogen. There are two types of caspase: initiator, it means 8, 9, 10, and executor, they are 3, 6, and 7. So these caspase are going to involve in DNA fragmentation like caspase 3, which are executor caspase, which involve activate caspase activated DNase. DNase is main role is break down the DNA by degrading the inhibitor caspase activator DNase. So CAD cleave the chromosomal DNA into 180 to 200 base pair. We already know, okay, this is the main hallmark for a caspase which will only break down the 180 to 200 base pair, and we visualize it into DNA in a gel. then it appear in a DNA ladder like pattern in agarose gel electrophoresis, right? Then another caspase, they are involved in breakdown of the cellular protein. In the cell, there is a nuclear lamine, it involved in nuclear breakdowns, cytoskeleton proteins like actin, keratin, fodrin, they are involved in cell, so that can cell shrink and membrane blebbing will be occur, as well as the poly-ADP ribose polymerase, it start to break down because it involved in DNA repair. But here DNA repair is not possible. That's why apoptosis process will occur. So this enzyme will not repair during apoptosis process. That while it will break down this kind of enzyme. So these are the role of caspase. Now let's discuss how which biochemical events occurs in the apoptosis process. So as we discussed, procaspases are inactivated zymogen proenzyme, which are going to activate the caspase. How it will activate, that already we discussed. Then after activation, it cleaves the cellular proteins like PRP. It will inhibit the repair the DNA, cytoskeleton protein, keratin, all that, and nuclear lamina. Then it also involve in activation of caspase-activated DNase, which will activate the CAD, and it involve in the as a endonuclease activity. So internucleosomal DNA fragment 180 to 200 base pair of the DNA and DNA ladder pattern on agarose gel. Then after chromatin are going to condense, and thus condensed chromatin are fragmented into nucleus. Various fragmented nucleus are occur in various part. Then after membrane blending, here you can see that plasma membrane form the blebs surrounding the membrane. Apoptic body formations cell break into membrane bound apoptic bodies, and after that phosphatidylserine externalization will be occur. Ideally this cell membrane are internal, this situation how it will occur. So here you can see this is a normal cell. In normal cell, this is a phosphatidylserine is present into internalize, but when apoptosis process will occur, and at that time this phosphatidylserine will going to externalize, so it will give the signal it me. That's why macrophages are come and activate and will be give the apoptosis process or phagocytosis process. And that after phagocytosis by macrophage, which engulf the small apoptic body, and upon no inflammatory response will be occur and apoptosis process will be occur. Apart from this, mitochondrial outer membrane are going to permeabilize in case of intrinsic pathway. So it regulate which kind of cellular this cytochrome C going to release, and loss of mitochondrial membrane potential, so result in decreased ATP production, and it will promote the cytochrome C release. So these are all are the biochemical events will be occur into the apoptosis process or biochemical events. So these are the various MCQ. You have to solve this question and give me answer into the comment box. These are the another question bank. These are related to this topic. Solid and score in your important exams. Thank you very much for listening this episode.